Hello students, welcome to Gnana Deeksha, concept-based digital learning. I am Ananth Aparna Sumanjali, lecturer in Microbiology, Telangana Social Welfare Residential Degree Colleges for Women, Mahbubnagar. In recent times, entire world is concentrating and constantly talking about this one word regarding health. Can you guess it? Yes, it is immunity. And we all know that our immune system protects us, right? It protects our body from harmful pathogens like virus, bacteria, parasites and other forms of infection. So that's why we can compare our immune system with the defense system of a country which protects us from all kinds of enemies. Immune cells act as guardians or army for our body, just like you are seeing on the screen. But have you ever imagined what happens if an army attacks its own country? The story I'm going to tell you is about when the immune system makes mistakes and the guards attack its own body it meant to defend. Sometimes the immune system stops working properly, and instead of attacking harmful invading antigen, it mistakenly attacks your own cells, resulting in a disorder called as autoimmunity. In today's class, we'll be learning about autoimmunity, what they are, what they do, and the facts you need to know about your body. By the end of the session, you'll be able to know what is autoimmunity, facts of autoimmunity, factors affecting them, types, that is classification, and mechanisms of autoimmunity and treatment methods. So let's get started. Autoimmunity is a condition where the immune system fails to differentiate between self and non-self. Basically, usually it differentiates what is self cells, what is non-self cells. Due to this failure of an organism, immune system starts recognizing its own constituent parts, that is self molecules, as foreign molecules, and it evokes an immune response against its own cells and tissues. For better understanding, let's look into this example. What do you see? It's related to a movie, right? Can you name it? Yes, this picture is from a famous science fiction movie named Robo. In this movie, a scientist creates a sophisticated humanoid robot named Chitty to commission it into the Indian Army. He prepares this humanoid robot to get deployed into Indian Army to protect its civilians. But the story gets a twist when the villain embeds a red chip inside Chitty while reconstructing it, converting it into version 2.0, which is more aggressive and unempathetic. This new version of evil robo starts creating more humanoid robo like him as an army of its own to fight against its own creator and other humans it originally meant to defend. In the battle between Chitty's robo army and the police personnel leads to a property destruction and many casualties. Now, let's imagine your immune system is like Chitty the robo. When it's normal, immune system functions well in protecting our body from harmful pathogens by attacking them. But when there's a glitch in your body, just like the red chip in the robo's body, when there is a glitch or sudden change in your genes, your immune system starts malfunctioning. Instead of attacking foreign antigens, it now starts attacking self molecules, and further it creates a group of such self-reactive cells, resulting in damage of our own body cells and tissues. Usually, the healthy immune system can tell the difference between self antigens and non-self antigens, basically what's you and what's foreign. Antigens are nothing but substances, usually proteins or carbohydrates, that has an ability to induce specific immune responses when they enter into our body. Just like our face is unique to us and people identify us looking at our face, at cellular and molecular level, antigens or unique markers present on the cells. Every cell in our body has markers that identifies it as being uniquely you. Organisms like bacteria, viruses have their own unique markers as well, and our immune system try to look for these foreign antigens to know when to attack. These antigens are of two types. First one, the antigens that are present on our own cells are known as self-antigens, while the antigens that do not arise or originate in our body are considered as foreign antigens or non-self antigens. Our immune system is designed in a way not to respond against self antigens. This is called as self-tolerance, whereas it strongly responds against non-self antigens. But in autoimmune disorders, immune system loses its tolerance towards self antigens and evokes a strong response against its own cells. Let's go back to the history. Going back to the history, we have Paul Ehrlich, a German physician and Nobel laureate. In his early immunological research, he wanted to know if the antibodies can be formed against body's own cells. So he experimented. In a series of his experiments, he and his colleagues tried to elicit the production of autoantibodies against RBCs in goat. Autoantibodies are nothing but special proteins synthesized by B lymphocytes. B lymphocytes are nothing but WBC, which act as immune cells. Whenever any foreign antigen enters into our body, these B cells detect them and get activated, and to fight against these, they release special proteins called as antibodies. But when these antibodies are targeted against our own cells, such antibodies are called as autoantibodies. So Ehrlich tried to detect the presence of these autoantibodies in the body. But after conducting a series of experiments, all of his attempts failed. Then Ehrlich started doubting the existence of autoantibodies and presumed that autoantibodies could not exist. And to explain this scenario, he coined the term horror autotoxicus, literally meaning horror of self-toxicity. Based on his theory, for over 50 years, 5 decades, scientists believed that the immune system was unable to react against its own cells. Later, in 1957, a German-American immunologist named Ernst Witebsky proved that autoantibodies were a true cause of autoimmune diseases. And he even demonstrated this by induction of autoimmunity in animal model suffering from chronic thyroiditis. Let's know the interesting factors and the facts about these autoimmune disorders. It's not just one disease, but a group of or a family of over 100 different types of autoimmune diseases known to the medical world that can affect almost any part of your body. Some common ones you may know include type 1 diabetes mellitus, rheumatoid arthritis that affects the joints, Hashimoto thyroiditis that affects the thyroid, etc. And statistics vary on how many people exactly suffer from autoimmune disorders, but approximately 10% of world population suffer from some or the other form of autoimmune diseases, and studies show that 1 in 5 American people suffer from autoimmune diseases, and these diseases can affect people of any age group. And studies have shown that autoimmune diseases are more common in certain ethnic groups. For example, lupus. Lupus is a type of inflammatory disease. Inflammation is nothing but swelling. This disease is seen to affect more in African American and Hispanic people compared to Caucasians. They are more common in women than men, as more than 75% of the patients who are suffering from autoimmune diseases are female. And studies have shown that young and post-pubescent women, that means the women who are in reproduction age, or shown 10 times more susceptible to diseases than men. So these are very incredibly important for women to be aware of. It's not entirely clear why women are more susceptible to autoimmunity, but some researchers speculate that women's ability to overproduce estrogen hormone may play a major role in it, because studies have shown that estrogen like molecules can alter immune response. Certain autoimmune diseases like multiple sclerosis, a nerve damaging disease, and lupus, just now we have seen lupus is an inflammatory disease, that these type of diseases are shown running in families. Not every family member will necessarily have the same disease, but they inherit a susceptibility gene to an autoimmune condition. Now let's get into the factors influencing autoimmunity. The root cause of autoimmune diseases remain a mystery to solve, but there are some connecting factors. There can be genes that are predisposed by inheritance. Predisposition is nothing but having them in your body already. Just now we have seen that autoimmune diseases can run in families, right? That means the tendency to develop these autoimmune disorders can be inherited. If you have family members who have autoimmune disorders, your chances of getting the same disorder or the one that is closely related are higher. There is some evidence that exposure to certain things in the environment may increase the risk of developing autoimmune disorders. Simply having the susceptible genes will not make you prone to the disease, but when those susceptibility genes get a triggering point, they will give you autoimmune diseases. These triggering factors also include irradiation, that is exposure to radiation, or exposure to toxic chemicals, or exposure to drugs. For example, research shows that exposure to some medications like procainamide and hydrolazine and certain metals like mercury, gold or silver may be associated with the development of autoimmune disorders. But there is no scientific evidence relating that environmental exposure to the development of autoimmunity. But still researchers are working to find out how these environmental exposure play a role in induction of autoimmune diseases. There is mounting evidence that genetically susceptible people who have had certain bacterial and viral infections may be at risk for some types of autoimmune disorders. But it is also still unclear exactly how these infections may increase the risk of autoimmune disorders. Children today aren't exposed to as many germs as they were in the past, right? The lack of exposure could make their immune system prone to overreact to harmless substances. So, lifestyle is also a triggering factor. Now, let's look into the classification of autoimmunity. Autoimmune diseases can be classified according to several criteria. One among them is based on the location of autoimmune attack. Organ-specific autoimmune diseases. These are the diseases where a single organ or a particular tissue is preferentially targeted by the patient's altered immune system. For example, the thyroid gland in patients with Graves' disease or Hashimoto's disease, or the pancreas in the case of diabetes mellitus patients. Organ-specific autoimmune diseases result in two outcomes. First is direct cellular damage, and the second is abnormal functioning. Abnormal function in the sense either it may overstimulate or block the normal function of those organs. Let's look into the first one. The first is direct cellular damage, where complete cell destruction of that particular target organ takes place. For example, autoimmune hemolytic disease. In this disease, the patient's immune system targets RBC of their own body and they make antibodies against red blood cells, resulting in anemia. The other outcome of organ-specific diseases include overstimulation or blocking of normal function of autoantibodies. Let's look at this example. This is a picture of a disease, autoimmune disease called myasthenia gravis. This is a neuromuscular disease leading to fluctuating muscle weakness and fatigue. Can you all raise your hand? Can you all twist it? Yes, you can do it because there are some signaling factors or signaling chemicals released in your brain telling you to fold your hand or raise your hand. This kind of voluntary movement is due to the release of neurotransmitters. In this myasthenia gravis, such kind of neurotransmitter called acetylcholine gets blocked, resulting in skeletal muscle weakness. As you can see on the screen, the main symptoms of myasthenia gravis is weakness in the voluntary skeletal muscles, which are muscles that you can control. The failure of muscles to contract normally occurs because they can't respond to nerve impulses. Without proper transmission of this impulse, the communication between nerve and the muscle is blocked, resulting in weakness in the muscles. Let's look into the second type of autoimmune diseases, that is systemic autoimmune disease or non-specific autoimmune disease. As the word suggests, non-specific, these diseases where the immune response is not specifically targeting one antigen, but it is directed toward a broad range of target antigens and involves a number of organs and tissues. For example, the cartilage or DNA protein complexes, which are spread all over our body. Consequently, the damage involves many different organs and tissues. Let's look at some of the typical systemic autoimmune disease examples. Rheumatoid arthritis, systemic lupus erythematosus, scleroderma, dermatomyositis. All these are systemic autoimmune diseases that affect at a broad range. These diseases are managed by special doctors called as rheumatologists, and in fact the term systemic autoimmune disease and rheumatic autoimmune disease can often used interchangeably. Let's look into example of systemic disease, that is rheumatoid arthritis. This is an example of systemic autoimmune disease in which your immune system mistakes the lining of your joints as foreign substances and it elicits antibodies against your own joint lining, resulting in severe swelling or inflammation and severe pain. This disease often affects the joints of the hands, wrist, knees symmetrically. Let's understand the mechanisms for induction of these autoimmune diseases. Following possible mechanisms have been proposed to explain the development of autoimmunity. They are failure of central tolerance, failure of peripheral tolerance, loss of immune regulation, release of sequestered antigen, neoantigen, cross-reactivity or molecular mimicry. Next is genetic factors. The last is polyclonal B cell activation. Let's know how these mechanisms work. Having a good understanding of tolerance gives us a better idea what happens in autoimmunity. Just like we get educated in our schools and colleges, the developing immune cells will also get educated or trained not to react against self antigens but to strongly react against foreign antigens, that is to perform self tolerance. After we get educated, to test how much we have learned, schools and colleges will conduct exams, right? Likewise, to ensure whether all developing cells have learned self tolerance or not, immune cells will get tested in two levels. The first step is known as central tolerance and the second step is known as peripheral tolerance. Let's look into the first step, that is central tolerance. Here the word central refers to the central lymphoid organs or primary lymphoid organs. These are the sites where the lymphocytes will originate and mature. The developing cells that detect self-antigens that bind them very tightly or effectively are considered as self-reactive or autoreactive cells. During the course of development, such cells which are detecting self-antigens are considered as autoreactive immune cells, and they are negatively selected, and they must be eliminated from our body. This is done by an important mechanism in the central tolerance by a procedure called deletion and apoptosis. Apoptosis is a programmed cell death where such self-reactive immune cells will be inactivated and they no longer be able to function or thrive. But the central tolerance is not foolproof. This is where the role of peripheral tolerance comes into play. Let's look into the second step, that is peripheral tolerance. Here, the term peripheral refers to the secondary lymphoid organs like spleen, lymph nodes, mouth, etc. Some of these autoreactive cells would have entirely escaped from the first test, that is from the central lymphoid organs. The main aim of the peripheral lymphoid organs is to capture these escaped cells and preventing autoreactive mature lymphocytes from attacking our own body cells. This is achieved by an important mechanism called energy. Now, let's know what is energy. Once the self-antigen encounters the lymphocyte, it is rendered or made functionally inactive. Usually, our T cells require two important signals to get activated. Signal number 1 is generated when the binding of T cells to antigen presenting cell happens via MHC molecules, and signal number 2 is generated when the T cell associated molecule, that is CD28 receptor, binds with co-stimulatory receptor, that is CD80. Unless these two signals are generated, autoreactive T cells will not get activated. So your body performs energy in stopping the second signal. By blocking the second signals, it results in inactivation of such self-reactive cells. Any failure in the deletion procedure of the first test, that is central tolerance, or failure in energy, that is the second test in peripheral tolerance, results in breakage of self-tolerance, causing autoimmune disorders. There is another important aspect, that is what is called as regulatory T cells. Regulatory T cells are small subsets of T cells which are devoted to lymphocytes activation or inactivation. Regulatory T cells have two major functions. The first one is maintaining self-tolerance in the body. The second is to suppress excessive immune responses which are deleterious to our body. Naturally, these T regulatory cells have two co-stimulatory receptors called as CD4 and CD25. Along with these two, they also express a protein receptor called FOXP3, that is Forehead Box P3 protein on their surface. This FOXP3 protein is crucial for the regulatory T cells for maintaining suppression of overactive immune responding cells. So, if there is any mutation happening to this FOXP3 receptors, it leads to the production of self-reactivity, and such self-reactive lymphocytes will start attacking healthy cells, resulting in autoimmunity. Let's look into the next autoimmune mechanism, that is release of sequestered antigens. We have just studied that our antigens which are exposed to the developing T cells during embryonic stage are only considered as self-antigens by the immune system, and to such self-antigens our immune system doesn't produce any immune response when they encounter them. But There are some self-antigens which are late developing in our body. For example, your own eye lens protein or sperm, etc. And these late developing proteins are normally concealed anatomically and protected from circulating immune cells. Any tissue or self-antigens that are not exposed to T cell during embryonic stage will not be tolerated by the T cells. Exposure of mature T cells to such normally hidden antigens at a later time might result in their activation. Example, the release of lens protein after an eye damage or release of muscle antigens after myocardial infarction has been shown to develop autoantibodies. The role of infection also has an important mechanism called molecular mimicry. In this mechanism, non-self antigens or foreign antigens will simply mimic our own self antigens. A number of viruses, bacteria have shown to possess peptide fragments, that means protein fragments which are similar to normal host cell protein fragments. So, the antibodies which are arised against these microbial antigens will now get confused. It doesn't know what to react. So, it will react both against the microbe as well as the self antigens. Let's look into the example. Immune system produces antibodies when a bacteria called streptococci enters into our body. This bacterial protein antigen shares similarity with some heart muscle antigen of our body, and the antibodies which are secreted against that antigen, foreign antigen, will now target or cross react with our muscle heart antigen and cause a disease called rheumatic fever. Another mechanism includes neoantigen or epitope spreading. As the infection continues in our body, some of the microorganisms will stay within our cells. These are called as intracellular microbes. On prolonged living of these microorganisms within our body can alter the cell surface antigens of our own body, and these microbes will expose those newly modified antigens to the immune system, resulting in targeting our own body cells, that is autoimmunity. The next and the last mechanism we are going to study is polyclonal B cell activation. Next is polyclonal B cell activation. First infection with viruses like HIV, Epstein-Barr virus have shown directly inducing the proliferation and differentiation of autoantibody secreting cells from different B cells. This phenomenon is known as polyclonal B cell activation. Basically, when an antigen enters, B cell will get activated and release signaling molecules to other B cells to proliferate. Okay, but in this case, the infections, due to the viral infections, the signals will come to the B cell which will proliferate and attack our own body cells, resulting in autoimmunity. Next off, we have treatment methods for autoimmune diseases. There is no cure for these autoimmune disorders. Once an individual gets it, they got it for lifetime. So, treatments can't cure autoimmune diseases, but they can control the overactive immune response and bring down the swelling or the inflammation and reduce the pain. Treatment depends on the disease, and they include hormone replacement, corticosteroids, non-steroidal anti-inflammatory drugs, DMARDs, biologics, plasmapheresis, splenectomy, physiotherapy, and stem cell therapy. Let's look into the first one, hormone replacement. In some autoimmune diseases, hormone producing glands are destroyed, leaving the body without the key parts of its messaging system. When this occurs, a doctor can prescribe replacement of hormone. For example, is the insulin hormone usage in patients of type 1 diabetes and thyroid hormone usage as a replacement in Hashimoto thyroiditis patients. The second treatment method is corticosteroids. These are the drugs which are used to control the inflammation by suppressing the overactive immune system. But there is a disadvantage with this treatment because it can lead to the body more prone to outside harm because they suppress whole immune system at the same time. Next treatment is usage of non-steroidal anti-inflammatory drugs which are used to ease the pain and swelling. The fourth one is DMARDs. DMARDs are nothing but disease modifying anti-rheumatic drugs which are used to reduce the pain and to suppress the overreactive immune system, and they also prevent long-term damage of joints and other organs. The next treatment is biologics. These are often called as immune response modifiers. These are classic drugs which will interpret the events that are driving the inflammation and specifically target them instead of targeting the whole body. Let's see another treatment method, that is plasmapheresis. In cases like rheumatoid arthritis, the combination of antigen and antibody will take place and seen all over their body. Such type of combinations are called as immune complexes. So plasmapheresis is a process where the liquid part of the body, that is plasma, is separated from the blood, and typically the plasma is replaced with another solution such as saline or albumin, or the plasma is treated and then returned to your body. This is just like dialysis for kidney patients. This is done to remove antigen antibody complexes from the body. Next is splenectomy. Splenectomy is nothing but removal of spleen surgically. This is one of the treatment options for hemolytic anemia. Next is physiotherapy. Physiotherapy is one of such treatments methods used for autoimmune diseases like rheumatoid arthritis, ankylosing spondylitis, multiple sclerosis to relieve pain. And next off is thymectomy. Thymectomy is one of the major treatments for myasthenia gravis. Just now we have seen an example. It is a surgical procedure where thymus gland is removed to stop producing autoantibodies that mistakenly attack the muscle nerve connections in myasthenia gravis patients. Currently, scientists are experimenting with stem cell therapy for autoimmune disorders. Such treatments involve reprogramming of damaged cells, or they'll completely replace the patient's immune system altogether by replacing the bone marrow, the major site where all these cells are generated. Since millions are suffering from autoimmune diseases, more new treatments and researches are available to keep the body's defense aimed towards its enemy rather than towards itself. Let's quickly recap what we've studied today. Autoimmune diseases is a condition in which your immune system mistakenly attacks your own body. This is due to the breakdown of special mechanism called self-tolerance, and we've looked into the facts how autoimmune diseases affects women more than men, and we've known that genes play a major role in autoimmunity and they can be heredited. These diseases are of two types: organ specific, where immunity is targeted into one organ, whereas systemic, where multi organs are targeted. And we also got an insight on various triggering factors like irradiation, post-infections, drug exposure, etc. And we've studied about various mechanisms that results in autoimmunity, and we've known that treatment can't cure, but they can reduce the overreactive immune responses by suppressing them or by reducing the pain which is caused due to autoimmune diseases. Thank you all.